Hello, dear friends. We are discussing about the paper number third, BO333, genetics and evolution. It's a paper number third. It includes the genetics and evolution. Question arises: what is genetics and what is evolution? First of all, we discuss about the genetics. Genetics is a branch of biology which deals with the study of heredity and the variation. The Sir Gregor Mendel is known as the father of genetics because of his work he devoted. He gives us idea about the inheritance by means of his work on the garden pea plants. Sir Gregor Mendel was born in 1822 and he conducted the breeding experiments in a garden pea which is botanically known as the Pisum sativum, and he studied the individual pea plants in a many generations. Then he put forth basic laws of heredity called as the Mendel's laws of heredity or the laws of inheritance. Therefore, Sir Gregor Mendel was also awarded as a Nobel laureate. He gives three laws as a laws of dominance, law of segregation, which is also known as the law of purity of gametes, and also the law of independent assortment. By means of his work, he find out that the like begets like. This theory which is put forth, and he proved the inheritance is transformed from one generation to next generation. In his experiments, he utilizes the garden pea plant, and the garden pea plant was selected on the basis of the 7 contrasting characters. The contrasting characters which are found out in his experiment is like as the dominant and the recessive characters in a garden pea plants, where the seed shape, seed color, pod shape, pod color, flower color, then flower location and the plant height. Like as the plant height as the tall and the dwarf, flower location as the axial and the terminal, then seed shape as round and wrinkle, seed color as the yellow green, pod shape as inflated and the constricted, pod color as green yellow, and the flower color as a purple and white. He utilizes these contrasting characters for his cross. In his crosses, he utilizes like as the monohybrid cross and the dihybrid crosses. He utilizes the single contrasting character for the monohybrid cross. Then hence we first of all discuss about the monohybrid cross. In a monohybrid cross, the cross which is between two individual parents that differ only in a pair of a contrasting characters called as a monohybrid cross, where he utilizes a single contrasting character for a cross. Then the F1 progeny which is obtained after crossing these single contrasting characters, they will be known as the monohybrids. It is also known as the heterozygotes. Generally, the monohybrids, those are obtained after the crossing between two different parents, that is the two characters. If we take the example like as capital TT crosses with small tt, that is the tall plants crosses with a small t, or the dwarf plants, then they will be obtained the F1 or the first filial generation, which is generally the heterozygotes. The phenotypic ratio of these offsprings obtained in F2 generation is as a 3:1, while the genotypic ratio of the F2 is 1:2:1. Then we'll explain about it. Take the two parents, one parent which is with the tall, that is the plant height is as a tall, and another as the plant height as a dwarf. Then when we take the plant height as a tall and the dwarf, then it is symbolized genotypically by means of this capital TT for the tall and small tt for the dwarf. Then both are the homozygotes. When we cross them, Then they produces the gametes as a capital T and the small t respectively. Then the F1 progeny which is produces after them, they will be after crossing them, then the capital T crosses with small t and the F1 progeny which is obtained, which is a heterozygote, as a capital T and the small t. Then these heterozygote, they appears phenotypically, that is externally they will be all are the tals. Then when we self them, that is the F1 progeny, when we cross with the F1 progeny, then they will be obtained the ratio which is in F2 generation, that is the second filial generation, as the capital TT, capital T small t, capital T small t and the small tt. That is, they will be appear as a 3:1 ratio. And hence it indicates from this ratio we can conclude that the law of dominance. How? Because of when we cross two different organisms or the two different parents, those are with genotypically dissimilar but both are the homozygotes, then they will produce as the progenies in F1 when they are appear like as a capital T small t. They will be appears as a dominance, then they will be phenotypically expressed as a tall. By means of this, they will be conclude that the law of dominance. Law of dominance states us that those are dominants, they will be expressed in F1 generations, and those are unable to express the other recessives. By means of this monohybrid cross, we can conclude that the dominants will be expressed in F1 generations and recessives are unable to express in F1 generations. Then it can be also explained like that the dominant character suppresses the expression of the recessive character, and those expresses they will be known as the dominants. Then the ratio which is obtained after crossing them, then the F2 progeny which is produces, they will be the like as the 3:1. 3 will be the tall, 1 is the dwarf, that is the dominant will be expressed in mode ratios. Out of 4 progenies, 3 progenies are the dominants and the 1 is as a recessive. This is about the monohybrid cross. Then by means of the dihybrid cross, in a dihybrid cross, Sir Gregor Mendel take one example. For to cross the dihybrid cross, he take the one parent which is with the two contrasting characters. Then what is a dihybrid? First of all, we discuss about it. A cross between the two pure or the homozygous individuals. The cross between two pure or the homozygous individual parents which differ in the two pairs of the contrasting characters is known as the dihybrid cross. Then, first of all, the F1 offspring resulted from the dihybrid cross is known as the dihybrids, and these dihybrids is always heterozygotes. Then the phenotypic ratio of these offsprings when we cross these F1 progeny again with the F1 progeny, that is the selfing, they will be resulted as the F2 generation, and they will be with the ratio like as the dihybrid ratio is as a 9:3:3:1. Then first of all, the cross between the two parents, two pure parents, two homozygous parents, two different parents which are with two different or the contrasting characters are taken. Such crosses are known as the dihybrid cross. These dihybrid cross resulted into the formation of the dihybrids, and the ratio is known as dihybrid ratio. For this purpose, Sir Gregor Mendel was selected the garden pea plant which is with the yellow round first progeny which is with capital YY, capital RR, and he crosses with the pea plant which is with green and the wrinkle seeds. Then when we take such a plants, those are with the homozygous. In this case, towards the left side we must have to write the male parent, towards the right side we must have to written the female parent. Such two seeds, such two parents when we selected, genotypically both are homozygous. They produces the gametes like as when the yellow round, which is symbolized by capital YY, capital RR, they will produces the gametes like as capital Y, capital R, and the another progeny which is with the green wrinkle seeds, they are with capital small y, small r. Then they produces the gametes as a capital Y, capital R, and small y, small r. After crossing them, they produces capital Y, capital R, small r. Such a F1 progeny, this is known as F1 hybrid. This F1 hybrid is expressed in the form of yellow round, which is heterozygote. It's a dihybrid, but they expresses like as capital Y and capital R, and hence here the capital Y and capital R are the dominants. Then after synthesis of it, the F1 progeny, when the F1 progeny again crossed as a selfing, then this selfing resulted into the progenies like as 9:3 3:1. We'll discuss it by means of the checkerboard, where they produces the gametes capital Y, capital R, capital R, small r. They will produces the gametes like as the capital Y, capital R, small y, small y, capital R, small y, small r. Then same gametes are produced by means of the female, and they after crossing them, they will produces such a progenies. Those are with 9 progenies with yellow round, 3 progenies with yellow wrinkle, 3 progenies with green round, and single progeny is with a green wrinkle. On the basis of it, the dihybrid ratio is as 9:3:3:1. Okay.